Hello my dear students, in this video we will explore on the topic how fishes balance salt and water. It is the process basically known as osmoregulation. So osmoregulation is the physiological process by which organisms maintain the balance of water and electrolytes in their bodies, essential for survival in environments with varying salinities. For fish, osmoregulation is particularly vital as they inhabit a wide range of aquatic environments from freshwater, river and lakes to the vast saline expanses of the ocean. According to their habit, fish can be categorized into two main groups: marine fish and freshwater fish. So in case of marine fish, the process of osmoregulation can be divided in two categories depending on their habitat. If the habitat is iso or slightly hyperosmotic, so in that group of fishes, example hagfish or elasmobranchs like sharks, rays, latimeria, etc., their internal concentration is more or less same with the sea water, so no major water loss or gain happens there, so minimal osmoregulatory effort is found. In case of hyperosmotic fishes, where examples are lampreys and teleost like bony fishes, their internal salt concentration is lower than sea water, so constant water loss occurs by means of osmosis, so no need to drink seawater and excrete excess salts. But in case of freshwater fishes, hyperosmotic, their body is hyperosmotic to their environment, so internal salt concentration is more concentrated than the surrounding water, so constant water gain occurs by means of osmosis, so no need to excrete excess water and actively absorb salts. A summary of various fish species along with their habitats and osmotic characteristics can be found in this table. Cyclostomes basically represents an early evolutionary stage in vertebrates. By studying the process of osmoregulation in cyclostomes, we can trace how more advanced fishes or teleost or elasmobranchs can evolve more efficient osmoregulatory systems. Cyclostome, who are basically jawless vertebrates, belong to the superclass Anatha, which is considered a part of the fishes in a broad zoological context. So in case of cyclostome, in case of hagfishes, which are strictly marine and stenohaline, they have narrow salinity tolerance. They are usually isoosmotic, so their body fluid salt concentration is equal to the seawater salt concentration. Their blood sodium concentration may be higher than seawater, so no major osmotic stress and minimal osmoregulatory effort is found. In case of lampreys, who are anadromous, who live in both freshwater and seawater, the body fluid osmolarity is approximately 1/4 to 1/3 of that seawater, and result they face osmotic stress similar to marine teleost. Their adaptation, they adjust osmoregulation based on their habitat. Osmoregulation in elasmobranchs, just like sharks and rays. In case of marine elasmobranchs, their body fluid salt concentration is approximately 1/3 of the seawater, so they retain urea to raise or to increase the total osmotic concentration, which is nearly similar to the seawater. So they are isoosmotic. So this urea retention is performed by the reabsorption with the help of the kidneys instead of excreting the urea. So here one problem can be arise. This retained urea can destabilize the proteins. So the solution of the problem is the using TMAO, trimethyl amine oxide, which stabilizes the proteins. Still, it regulates ions like Na plus and Cl minus, which diffuse in through gills and food. Excess salts are excreted by kidneys and rectal glands, which are main organ for salt excretion. Water enters slowly by osmosis and it is used for urine and gland function. Freshwater elasmobranchs, examples are bull shark and Amazon stingray. Their lower body salt and urea levels than marine species. The reduced urea, that is less than 1/3 of the marine sharks, and less water inflow. Kidneys excrete less water and they conserve solutes. It is unclear if adaptation or passive effect, but helps survival in freshwater. So this is the diagram of osmoregulation in elasmobranch. Now, we should know what is the role of TMAO in fish osmoregulation. TMAO is basically trimethyl amine N oxide. It's a special compound found in many marine or saltwater fishes. Its main function in osmoregulation is to protect the fish's body cells from the harmful effects of salt and pressure in the ocean. It works simply by salt stress protection in salty sea water. Fish can lose water from their body cells and take in too much salt. TMAO helps balance the salt levels inside the fish so their cells do not get damaged. Protein protection. High salt levels can make proteins in cell stop working properly. TMAO helps keep protein stable and working even in salty or high pressure conditions like deep sea environment. Osmolyte function. TMAO acts as an osmolyte, a substance that helps maintain the correct water balance inside the cells, preventing them from shrinking or swelling too much. Osmoregulation in teleost or bony fishes. Osmoregulation in teleost live in marine and freshwater environments. Some species which can migrate between saltwater and freshwater and adjusted their osmoregulation accordingly. In case of marine teleost, they have hyperosmotic body which has less salt than seawater. They lost water by osmosis mainly through gills. They drink seawater to replace lost water. Excess salts are excreted through gills via chloride cells. The kidneys produce only dilute urine which cannot concentrate salts. Salts removal is an energy consuming process. Freshwater teleost hyperosmotic body has more salt than water. They gain water by osmosis mainly through gills. They produce large amount of dilute urine to remove excess water. They lose salts in urine. To retain salts, they actively absorb salts through gills and also get some salts from the food. Skin plays minimal role in osmoregulation. Here is the diagram showing that teleos osmoregulation in seawater and teleos osmoregulation in freshwater. So, we can conclude that osmoregulation in fish is a vital physiological process that enables them to survive and thrive in diverse aquatic environments by balancing water and salt levels through specialized adaptations. So, thank you, my dear students, and all the best for your attention and curiosity in exploring this fascinating topic of fish biology.